We're going to take a look quickly at S phase, how we're going to do chromosome replication. Now, we've already looked at in previous chapters how the bases, those nucleotides, match up in DNA, and that is incredibly important when we go to replicate chromosomes. So during S phase, what's going to happen is a series of steps in order to make two identical strands, two identical copies of that DNA, so that each cell is going to get a copy of that chromosome that's identical to each other. So how does this work? Well, we've got the original chromosome, the original DNA molecule, and what's going to happen is that's going to get separated into two what we call template strands. Basically, these are going to be used as the original versions that we can build off of. The idea here is that basically, instead of trying to make a copy by looking and then going over and writing and then looking and going over and writing, you have the copy right there and you can write right next to it, right? You're building off of it. So that's exactly what happens. These strands are going to open up, and then we're going to just match the nucleotides straight to them. So you can see here, it's opened up, and so we're going to match the A to the T, A to the T, C to G, C to G, and the same thing's happening on the other side, right? It's just going the other direction. So A to T, A to T, G to C, C to G, T to A, then A to T. And so what we end up getting at the end are two identical DNA molecules, or two full chromosomes, right? This is only a little tiny section, but we would be doing this for the whole chromosome. And one side on each of these is the original molecule, and then the other side is built out of brand new nucleotides. Okay, so this is going to happen to every single chromosome in the cell during S phase. So just to remind us, refresh us a little bit, I know you're probably getting tired of seeing this, but I told you it keeps coming up. Which of the following is going to complement, so which of the following would be paired with this DNA sequence? So if, that was being written to make that new strand of DNA, which one of these would match with it? And in this case, it would be B, right? Because you would go A to T, G to C, T to A, C, G, and so on. All right. So how is this replication going on? We're just going to look at it at a very high level, very simple version. There's more details in your book if you want to take a look at it. We're just going to look at the overview. So basically there's two steps, right? The first is we need to open up and unwind that DNA. Remember that the DNA is in a double helix, so it's in that spiral shape. And we need to open up the two sides of the ladder because we're going to use those two sides as our template. So there is what we call DNA helicase. Ends in ASE, so that means it's an enzyme. In this case, it's an enzyme that untwists and unzips the DNA, right? So it's going to chug along and do that. Then following along behind it are DNA polymerase with a couple of little helpers. And this DNA polymerase, again, ACE ends in ASE. It's an enzyme. In this case, it's an enzyme that adds nucleotides. And so it's going to come along and pair up the correct nucleotide with the one on the DNA strand. Okay? And so you can see they're going in different directions. That's just because of the way that the DNA is built. But they're going to build these strands up until the whole chromosome is copied. Right? So step one, unwind and unzip with the helicase. And then the second part is to reconstruct. So to build the other half of each side of that DNA. Once that's done, we'll have the full copy of each of the chromosomes to be able to do the division later on. Now, if you remember, talked about animal chromosomes are linear. They're strings, right? They're straight lines. So why, whenever we see them in pictures, do they look like the letter X? My guess is that you've noticed that. So what is going on here? Let's take a look. Oop, no, go back. So this is what the chromosome looks like during normal, regular part of the cell cycle. So during G1, chromosomes look like strings. They look like, I told you before, they look like a ball of spaghetti in the nucleus. Okay, so they're all uncoiled, all spread out. So they're not really visible to us except for as like this big solid ball. But after DNA replication, so after this process that I was just talking about, what you've got are two of these strands. We call them chromatids. They're still both chromosomes, but they're sister chromatids because they are copies of the same chromosome. Okay? And they are connected together by a centromere. And so this is now what we count the chromosome numbers by, is by the centromere. So this is still counted as one chromosome, even though there are two copies there. So there's two chromatids, but one chromosome because there's one centromere. This is why they look like X's. You can start to see that shape. As the cell starts to prepare to divide, these chromatids start to condense farther and farther and farther until we get the X shape that you've seen in the pictures. And hopefully it makes sense that this is a lot easier to move around than this, right? And that's exactly why they condense, because this is not going to tend to get tangled up and get caught on other chromosomes during the division. This is going to be a lot easier to move around and not get stuck on stuff, right? Okay, so that is DNA replication, but you might be thinking, yeah, but we've talked about mutations, and mutations can happen during that, right? And yeah, this is one of those times that mutations can happen, right? Because a lot of errors can happen during this replication process. Just like when you take notes or copy something down, your replication machinery can make mistakes too. But you do have a bunch of different DNA repair processes that occur during and after replication. So that DNA polymerase actually has its own spell check. So it's actually making sure it's pairing up the right ones. Now, it does make mistakes. And so every once in a while, it will make a mistake. But there are other DNA repair checkers, other proofreaders that will go back in and check to see if there are mistakes. Because if you pair the wrong bases together, so for example, it's supposed to be A to T, apples to trees, right? But if you pair an A and a C together, they can't actually bind together. They can't bond because the bonds don't line up. And so when that happens, you get a bump in the DNA. And so the proofreaders can come along and see, oh, that's not right. This is supposed to be straight. And so they'll cut out the base that is sticking out and put in the correct matching one. Now, there are mechanisms to help tell them which one was the original one, but they don't always get it right. And so that's how sometimes you can get those mistakes. Plus, they don't always, catch all of the mistakes. So why is it that we can get errors be a good thing sometimes? So why are errors during replication possibly a good thing to occur? Yeah, like we've talked about before, mutations can be a good thing because we need mutations for evolution, right? All right, so next section we're going to head into is actually talking about mitosis. So the processes and reasons for mitosis.